DNA stands for deoxyribonucleic acid, and for the most part, it's found in every cell of our bodies. DNA is the material we inherit from our parents, and it remains the same from the cradle to the grave. Because of this fact, DNA can be compared and contrasted like a system of checks and balances. On account of such, when testing DNA, a DNA analyst should never come to a conclusion that is not verified, because DNA evidence can always be compared to its original source or sample. It is important that all DNA analysts follow the same protocols regarding the interpretation of DNA evidence, since more than 98% of our DNA is the same amongst all human beings. For example, most humans have 10 toes, 10 fingers, 2 legs, 2 arms, and 2 eyes, because we all share the same genetic makeup in those regards. When analysts specifically verify a particular person's DNA, they focus on 13 to 15 targeted areas of the DNA cell called non-coding regions. The cells within these non-coding regions, also known as loci, separate one person from another and help DNA analysts tell the difference or identify separate human beings. For example, when DNA tests are conducted and a DNA analyst can match all 13 to 15 targeted areas of a DNA sample, a mathematical calculation can determine if the evidence will include or exclude a particular person. This is how scientists and analysts are able to distinguish or separate the innocent from the guilty. Understanding the DNA process of how DNA testing is conducted. Step 1, DNA digestion. Step 2, DNA extraction. Step 3, DNA quantification. Step 4, DNA PCR amplification. Step 5, genetic analyzer run. Step 6, electropherogram statistic report. Step 7, DNA report. Step 8, testimony. It is important to note that steps 1, 2, and 7 are conducted by hand. Steps 3 through 6 are largely machine or computer aided, and step 8 is a verbal testimony given in the court of law. At this moment, let's direct attention to how an innocent man, Cyrus Casby, was framed for murder by the Jefferson Parish Crime Lab because steps 1 and 2 were improperly performed. Step 1 is the DNA digestion. It requires that a DNA analyst break open the DNA cell, concentrate it to actually get to the data inside. This step is vital and should only involve the DNA samples from one person at a time to avoid contamination or mixing between samples. Step 2 is the DNA extraction. After breaking open the DNA cell, analysts concentrate and purify the data found inside the DNA cell. This process makes use of a specialized oven at 132.8 degrees Fahrenheit, a fixed angle centrifuge, a microfuge, and many other testing materials. This step must also be a single DNA sample to prevent contamination or mixing. This step is recorded by date and time on Form SOP 205. It is important to note that steps 1, 2, and 3 need to be performed in a controlled and isolated environment using single DNA samples only. DNA from multiple donors should never be present nearby or exposed together in a laboratory during these steps. Step 3 is DNA quantification. After extracting the DNA from the cell, DNA analysts quantitatively or numerically determine how much DNA is present. This step is necessary because DNA analysts require a sufficient amount of DNA in order to run a successful test. Quantifying DNA is conducted by placing DNA samples on a quantitation plate and then inserting the DNA inside an ABI Prism 7000 SDA machine. This step is recorded by date and time on Form SOP 260. Step 4 is DNA PCR amplification. Once DNA analysts confirm that a sufficient quantity of DNA is present, analysts must amplify or clone this original sample so they can have duplicates of DNA to conduct testing. This cloning is carried out by using an amplification kit, a thermal cycler, and many other testing materials. This step is recorded by date and time on Form SOP 310. Step 5 is the genetic analyzer run. After the DNA is successfully cloned or amplified, the cloned copies are placed on a capillary plate and then put inside a machine called a genetic analyzer until testing is complete. This step is recorded by date and time on Form SOP 440. For testing, the Jefferson Parish DNA Crime Lab used an ABI Prism 310 capillary plate and an ABI Prism 310 genetic analyzer and many other testing materials. Step 6 is the electropherogram statistic report. After the genetic analyzer completes testing on the cloned DNA samples, it makes the results visible in graph form. These graphs are known as raw data, which display DNA peaks called alleles. Alleles are the name given to pieces of data found within 13 to 15 loci. Analysts use the information from these raw data charts to produce their DNA reports. Step 7 simply consists of the authored DNA reports. Based upon the alleles present in the electropherograms, the analysts compare the known evidence samples in steps 1, 2, and 3 from a suspect or victim to the unknown DNA samples or crime scene evidence. From these electropherograms, a DNA report that includes or excludes a suspect is produced. Finally, step 8 is the testimony. After including or excluding a suspect in the DNA report, analysts testify in court according to their reports. And as we know it today, these are the steps that encompass the DNA testing process for all crime labs nationwide. As was disclosed earlier, Step 1, DNA digestion, and Step 2, DNA extraction, are performed by hand. During these steps, it is critical that DNA analysts keep the suspect's DNA samples separated from the victim's DNA samples due to high risk of contamination or mixing of different DNA samples. This practice is so important that experts petitioned that Congress create a basic DNA quality system nationwide for all U.S. laboratories to follow. A list of quality safe protocols were enacted to ensure that DNA analysts are highly trained and adequately competent. These protocols were officially enacted in 1994 under Title 42 USC 14131, known as the Quality Assurance Standards. It was during the creation of these guidelines when a group of scientists concluded that not only should suspect and victim DNA samples never be tested together, but DNA samples from different donors shall be tested in separate spaces and also at different times during the initial DNA steps 1, 2, and 3 to help prevent the possibility of DNA contamination or mixture through use of equipment. However, in the case against Cyrus Casby, when DNA analysts of the Jefferson Parish Crime Lab were performing Step 1, breaking open the DNA cell, DNA analyst Greg Harrell digested and handled Cyrus's clothing together with the DNA of all the victims and on the same day, a violation. Not only was this conducted on the same day, but it was conducted at the same time frame, another violation. Cyrus's clothing was improperly digested and handled against procedures with the DNA of all the victims on May 2nd, 2005 at 11 a.m. This step is recorded by date and time on 2 JPSO forms, SOP 205. Additionally, during Step 2, DNA extraction, analyst Greg Harrell extracted DNA from Cyrus's clothing while simultaneously handling DNA of all the victims at the same place and at the same time. This is an egregious violation of scientific protocol. This DNA extraction took place on May 3rd, 2005, beginning at 12:45 p.m. and ending at 3:45 p.m. This is also recorded by date and time on 2 JPSO forms. Both of these acts violate the federally mandated quality assurance standards and expose DNA samples from both the suspect and the victims to cross-contamination and mixing. Subsequently, during Step 3, DNA quantification, DNA analyst Greg Harrell quantified Cyrus's clothes and the victim's DNA on the same 96 well plate, then placed the contaminated samples together to be tested inside the ABI Prism 7000 SDS. This is an extreme no-no. This was done on May 4th, 2005 on quantitation plate number Q050405 and is documented on 9 pages of JPSO forms, SOP 260. This mixture and contamination cannot be reversed because the evidence was contaminated prior to Step 4, DNA PCR amplification, also known as cloning of the DNA. Recall that it was earlier stated that Step 4, DNA PCR amplification, is when DNA analysts clone the original DNA samples. The analysts are supposed to preserve the original samples and run tests on the cloned DNA samples. If any mixture or contamination is created before this cloning process in Step 4, samples are registered incorrectly as original samples. But in reality, they are not true original samples. They are contaminated samples mistaken for true or original DNA samples. When the computer incorrectly acknowledges the contaminated DNA sample as an original sample, it helps distort the evidence. In other words, because of the DNA misinformation that the computerized graphs incorrectly outlined during the DNA cloning in Step 4, Cyrus's DNA results were contaminated with the blood of the victims. This gave the false appearance that victims' DNA was actually on Cyrus's clothing. But in scientific reality, DNA analyst Greg Harrell inadvertently contaminated Cyrus's clothing during Steps 1 and 2, DNA digestion and DNA extraction. This contamination allowed the computerized process to falsely acknowledge the contaminated DNA during Steps 3 and 4, DNA quantification and DNA PCR amplification. Moreover, DNA analyst Greg Harrell and Bonnie DeBorg conducted Step 4, DNA PCR amplification, with Cyrus's clothes and the DNA of the victims on the same day, a clear violation of procedure. This was recorded by date and time on 4 JPSO forms, SOP 310. In total, JPSO Crime Lab collected approximately 100 items of DNA evidence from the crime scene that contained human blood. Coincidentally, the only DNA samples that connected Cyrus to this crime were the samples of clothing that Greg Harrell improperly tested and contaminated with the DNA of the victims. Cyrus's contaminated clothing were the only pieces of evidence allegedly linking him to this crime. Without these contaminated clothing, there would be no other DNA found inside the crime scene that matched Cyrus. However, blood from an unknown male and an unknown female were found on both murder weapons and other places throughout the crime scene. The JPSO Crime Lab ignored and concealed those findings in the DNA report and from the court during testimony to frame Cyrus Casby. Testimony. The process to frame Cyrus was very simple. The Jefferson Parish Sheriff's Office arrested him in 2004. He was prosecuted by the state of Louisiana and acquitted in 2008 after a 5-day trial. He remained free for only 3 years until 2011, when the federal government re-arrested him for the charges related to the same crime. This second time around in federal court, DNA analysts would commit perjury to frame him and secure his conviction 2 years later in 2013. For example, after DNA analyst Greg Harrell contaminated evidence during the DNA testing process, in federal court, DNA analyst Bonnie DeBorg helped to conceal Greg Harrell's contamination by testifying that all of Cyrus's clothing tested positive for Janice Carto's blood, one of the victims. Her testimony in federal court was different than her first testimony in state court. In Cyrus's 2008 state trial, DNA analyst Bunny DeBorg openly admitted that blood from an unknown male and Janice Carto was found on Cyrus's clothing and shoes. However, later in Cyrus's 2013 federal trial, Bunny DeBorg testified that this same evidence only had Janice's blood on it. DeBorg never mentioned the existence of the unknown male. She intentionally withheld that the unknown male's blood was present and that it did not match Cyrus. This act of perjury and the concealment of contamination of evidence in this manner helped to wrongfully convict Cyrus to a life sentence in prison. On January 6, 2014, he filed a complaint with the U.S. Department of Justice. On January 9, 2014, with the local FBI. On February 5, 2014, he filed a motion alerting the judges in federal court. As a result, his own lawyer, Michael Rillman, and Judge Fallon ordered him to undergo a psychiatric evaluation. In April, he was shipped to Colorado to be evaluated. In June, the psychiatrist deemed him competent. From then on, he has filed complaints on November 29, 2014, with the Judicial Conference in Washington, D.C., on June 22, 2015, with the Louisiana Supreme Court, on September 30, 2015, with Amy Hess of FBI's headquarters in Washington, D.C., on April 11, 2017, with the Inspector General in Washington, D.C., and with the JSPO's accrediting laboratory, ASCLD. On May 11, 2017, with the Solicitor General in Washington, D.C., on June 26, 2017, with the U.S. Supreme Court, in August 2017, with the Jefferson Parish Sheriff's Office DNA Laboratory, the U.S. Attorney's Office, and 5 magistrate judges. On August 24, 2017, with the Louisiana Attorney General, on September 1, 2017, with Marjorie Eisman of the New Orleans ACLU, on September 14, 2017, with the Innocence Project of New Orleans, on September 27, 2017, with the Southern Poverty Law Center, and on October 24, 2017, with the Innocence Project of New York. If you're wondering why justice is not taking place in the face of this corruption, it's important to note that Cyrus, along with the forensic analyst department head at Southern University of New Orleans, Dr. Pamela Marshall, only first figured out the depth of contamination and the corruption in 2014. This is well after Cyrus's trial and sentencing in 2013. Since his imprisonment, Cyrus has reached out to multiple entities to expose the corruption and miscarriage of justice. None of these entities have responded favorably or within the confines of the law. The public is his last hope to expose this truth and for his freedom before his appeal is exhausted.